On November 26th of 2018, Dr. He Jiankui, a Chinese scientist, publicly claimed the birth of two twins as the very first ever gene-edited humans in the world. Specifically, Dr. He and his team used CRISPR-Cas9 genome-mediated therapy to delete the functionality of a gene called CCR5 in the human embryos. Now, CCR5 is a gene that in us humans codes for a protein that is found on the surface of our CD4 white blood cells that HIV uses as part of its infection pathway to make us sick with HIV. Now, Dr. He's research did not sit very well with scientists across the globe. Basically, the scientific community came out as a whole and condemned Dr. He and his research, and that is due in large part to the fact that using CRISPR on human embryos that would be implanted into a woman and then carried to term as a full pregnancy, giving a live birth, is considered a massive violation of internationally accepted ethics principles on human experimentation and human rights law. It's also downright illegal to practice in the United States. Now, in the days following Dr. He's announcement, he did attend the second International Summit on Human Genome Editing held in Hong Kong to answer questions from these scientists that were very curious and very skeptical of his research. But even though he did attend, the answers that he offered to the very critical questions by scientists were less than reassuring. While Dr. He was very quick to argue that there was a medical necessity for this experimental procedure to protect the daughters from inheriting HIV from their father, scientists were very quick to point out that the methodologies we use today in vitro fertilization techniques to conceive a child already result in the semen of a male undergoing what we call sperm washing, taking the sperm cells and isolating them out from the rest of the seminal fluid. This would then protect the daughters because as you isolate the sperm cells, you would be separating them from any HIV particles that would be able to infect the offspring. And this methodology is tried and tested and safe, and it's something that is clinically used in humans today. And beyond that, we don't currently have a very strong scientific understanding of the off-target mutations that could occur when we subject a human genome to CRISPR-Cas9 genome therapy. In other words, this man wanted to use CRISPR-Cas9 to delete a very specific gene, CCR5, but we don't have a thorough understanding of what other consequences might occur if CRISPR-Cas9 were to also induce mutations in other parts of the genome. And even within the gene CCR5, we don't necessarily have the fullest understanding of what the long-term consequences will be of deleting that protein that would normally be found in Chinese infants and teens and adults. So as these girls grow up in their lifetimes, there are calls saying they might be more susceptible to contracting influenza or West Nile virus and other complications we might not even really be aware of yet. And beyond the sperm washing debacle, it is also believed by scientists that there was a substantial lack of informed consent. In other words, letting the couple know about other alternative options they might have had to conceive these daughters beyond using the CRISPR-Cas9 technology to have a safe birth from HIV infection. And it is very obvious that there was an abundant lack of transparency as Dr. A conducted most of his research in secret. And pushing science aside for just a moment, it seems that Dr. He hasn't really considered the lives of these girls, and conducting this experimental procedure on human embryos is going to result in human babies that are going to grow up and have actual lives. And it seems like he doesn't really have much in terms of a long-term plan for supporting the well-being of these children as they grow up and learn about their nature as the first edited humans in the world. Now, the reason that I kind of feel like this is important for me to be talking about is because CRISPR is a technology that I fell in love with in college, and it is something that I studied extensively, and it's a technology that I do believe could have practical impacts in curing currently untreatable diseases that are genetically inherited, like Duchenne muscular dystrophy or Huntington's disease. But when scientists like Dr. He go rogue and misuse the technology in a race to be the first scientist to do something revolutionary and world-changing, even at the cost of circumnavigating ethical board principles, you can lose the trust of the public. And as the public is hearing this news story about mutant, crazy, gene-edited humans by Dr. Hay's CRISPR technology, it can result in a public distrust of CRISPR as a whole, and that could be bad for scientists who are seeking funding to do legitimate research using CRISPR-Cas9 genome therapies. I actually once made a video myself on using CRISPR-Cas9 to treat HIV in human patients, which maybe sounds like it's almost the exact same as what Dr. Hay has gone ahead and done, but what you should know is that in the video that I was discussing, that involved taking an adult human who is HIV positive, extracting a sample of their CD4 white blood cells, editing those white blood cells, or body cells, in vitro in a dish to delete the CCR5 protein, and then putting those white blood cells back into the patient so that they can proliferate in that patient's body and be immune to the HIV infection. Now, in theory, that experiment would result in that specific individual developing, through gene therapy, an immunity to HIV infection. That is something that would not be passed on to that individual's offspring, though, as that is not a gene edit that would occur in the human germline. And even in that case, where we're only editing body cells, that is still something that is not happening in humans yet. Even that is something that is very experimental. The study that I was citing was still looking at these things in mouse models. So just try to remember that in the wake of this scientist who acted in a horribly irresponsible way of misusing the technology and violating ethics policies, the technology itself, CRISPR-Cas9, is an extremely versatile, cheap, and easy to use new method of gene engineering that allows scientists a much greater degree of freedom to treat various genetic conditions that we didn't think would be this possible in the past. Thank you for watching, and stay inquisitive.